Hello everyone, I'm Huang Di. I was a PhD student of Professor Zhou from 2001 to 2005. By then, Professor Zhou and I proposed some idea to analyze the huge data and apply those ideas to microarray data. Microarray containing the expression of the tens of the thousands of genes. So it is the high-dimensional data, and it is challenging for some conventional algorithm and is good platform to test our idea. Those studies gradually steer me into the area of the bioinformatics. In 2007, I came to the United States of America working as the bioinformatics postdoc. And in 2010, I moved to a computational branch of the National Institute of Health. Now I'm working on the gene regulation. Before briefing you on one of my projects, I'd like to work through basic biological ideas. Basically, in a living cell, all the life information is encoded along the DNA sequence. In response to the external and the internal stimuli, the DNA sequence is transcribed and then translated into the protein products, which determine the cells, the function, the fate, and the behaviors. So there are two types of the DNA sequence. The DNA sequence carries the protein information, which we call gene, and the DNA sequence, which carries the information to determine when, how many, which protein is produced, which we call the DNA regulatory sequence. In a human cell, there are 20 to 30,000 of the genes and at least hundreds of the thousands of the regulatory elements. Among those regulatory elements, there are millions of the small pieces prone to the alteration across the different individuals, which make everybody unique and the whole human population nuanced. Unfortunately, a very teeny tiny fraction of this alteration are responsible for the disease. So to find out those disease-associated alteration is like to searching for a needle in the haystack. One of my projects is to find out the DNA alteration responsible for the liver disease. In this project, my team collected the information, DNA sequence information of thousands of patients and normal people. After that, we identified the DNA alteration or DNA sequence which can distinguish the patients from the normal people. Focusing on the DNA alteration or DNA sequence, we extract the information of features which are unique to the DNA sequence or DNA alteration. And we hypothesize that those features are responsible for the liver disease development. And we ask, finally, our biologist collaborator design the experiment to test our hypothesis. So this is basically how our project are carried out. Here, I just give you a sneak peek of the bioinformatics project. Hopefully, I've gave you some inspiration for the bioinformatics, which are very promising R&D area. Thank you.